Assalamualaikum warahmatullahi wabarakatuh. My name is Erma Aisyafika Binti Sharuddin, a final year student in Bachelor of Science Biology at UiTM Jengka, Pahang. Welcome to my presentation. Today, I'm going to present a final year project entitled Phytochemical Screening, Phenolic Content, Antioxidant and Antimicrobial Activity of Clitoria ternatea, supervised by Ms. Nur Tabita Binti Shaikh Nasir. These are the contents for my presentation. Background of study. Clitoria ternatea, which is oftentimes known as butterfly pea, comes from the Fabaceae family. It has bioactive compounds which act as favorite sources of medicine and has active defense systems such as antimicrobial, antioxidant, and many other pharmacological effects which can be obtained from every part of these plants. Next, the significance of study. The analysis of these plants as natural treatments to treat incurable disease has resulted in tremendous requests for therapeutic plants. Therefore, the phytochemical screening, antioxidant, and antimicrobial activity can draw a possible mechanism of action of the C. ternatea that helps the improvement of the pharmacological industry by new development or formulation of drugs into effectual medicine, which major results will aid the society. The problem statement. Infectious disease from pathogenic microbes cause minor to major human illnesses, so therefore the treatment options may be limited. Next, the objectives of the study are to discuss the chemical constituents, antioxidant activity, antimicrobial and antifungal activity of C. ternatea. These are the research questions. Morphologically, this plant is a perennial trailing climbing herb which rise from a woody rootstock. It has corollae that help protect the floral organs, and the petal shape is a funnel shape which can grow up to 5.5 cm long. On the next figure, its fruits are linear oblong pods with gleaming appearance of seeds. Ecologically, the plants can grow in a variety of soil types ranging from sandy to deep alluvial, loams, and heavy clays. Traditionally, C. ternatea was used to expel toxins from the body and to relieve body heat such as in fevers. The ascetics, swelling of the abdominal viscera, sore throats, and skin ailments were all treated with root of the C. ternatea. The seeds and leaves were frequently used as a brain tonic and improve intellect as well as memory. There are various types of chemical compounds that can be found from different parts of C. ternatea through phytochemical screening. 7 compounds were detected by GCMS. The highest peak error in correspond through the GCMS recorded was 48.77 by N-hexadecanoic acid and the lowest was 2.80 by N-decanoic acid. N-hexadecanoic acid is known as palmitic acid, which very useful to produce energy in human body and perform biological functions. But recently, GCMS analysis in extract of C. ternatea revealed a complicated mixture of 24 distinct chemicals. Among the chemical compounds that can be found are the phenolic content, flavonoids, terpenoids, tannins, saponins, and ascorbic acid. They exhibit beneficial functions that may work on human bodies and be the alternatives for certain existing drugs such as natural antioxidants, anti-inflammatory activity, anti-cancer activity, anti-depressant activity, and many more. However, some of them are at disadvantages if consumed below or over the suggested amount. For example, the saponins. It suppresses digestive enzymes and interfere with steroid action and absorption. Next, the significance of C. ternatea are the method that can be used to detect the presence of antioxidants are the FYP assay, DPPH assay, the reducing activity assay, and the hydroxyl radical scavenging activity. The extract of C. ternatea from flower may protect canis erythrocytes caused by AAPH from hemolysis and oxidative damage. Erythrocytes treated with the same extract of C. ternatea had significantly lower AAPH-induced lipid peroxidation and protein oxidation levels. The C. ternatea flower extract showed another significant effect in reducing the blood glucose level, as it consists of chemical compounds that inhibit pancreatic alpha amylase and intestinal alpha glucosidase activity. From the aqueous leaf extracts of C. ternatea, It comprises of enzymatic and non-enzymatic antioxidants. They are superoxide dismutase, catalase, glutathione peroxidase, and vitamins, respectively. Next, the antimicrobial activity. The leaves part has demonstrated to have strong antibacterial activity against E. coli and V. cholerae, known to cause dysentery. The ethanolic extract of C. ternatea root are effective against both gram-positive and gram-negative bacteria. This extraction had the highest potential, while the water extract had the lowest antimicrobial activity against 3 bacterial strains, E. coli, S. aureus, and P. aeruginosa. The crude extract from C. ternatea had the highest zone of inhibition against E. coli at a concentration of 0.75 mg and the lowest against the amphlebus. The C. ternatea petals extraction yielded the highest content of anthocyanin and demonstrated the highest antibacterial activity, which inhibited the oral bacteria of S. mutant. Next, the antifungal activity. C. ternatea's ethanolic leaf extract treatment was reported to minimize the increase in activity of the stress enzymes caused by the fungal infestation. The seed extract of C. ternatea also displays significant antifungal activity, with a minimum inhibitory and fungicidal concentration against Aspergillus niger and Aspergillus ochraceus. The extract showed promising antifungal activity. The phenolactin obtained from the seeds of the C. ternatea also exhibit the same properties. C. ternatea also exhibit the cytotoxic activity, in which it can be demonstrated from the extract of C. ternatea leaves, seeds, and stem bark. It is also safe to be used as a natural coloring agent because some consumers may have allergic reactions and affected by the toxin synthetic coloring. It also exhibits the anti-asthmatic and antihistaminic, which the ethanol extract of C. ternatea roots reduces the bronchial hyperactivity and protects the rest from histamine-induced bronchoconstriction. C. ternatea also can treat Alzheimer's disease by acting as a therapeutic agent. And not just that, the extract of C. ternatea also demonstrated to have antioxidant efficacy in the gastrointestinal activity. Next, the C. ternatea seed and root extracts have similar action of wound healing properties with the cotrimazole ointment. For anti-anxiety effect, the methanolic extract of C. ternatea had a dosage-dependent anti-anxiety effect in the mice, which showed that the extract exhibits a mild sedation and muscle relaxation. This showed that the plants have the effect to suppress the anti-anxiety activity. Next, the anti-helminthic effect. The C. ternatea flowers, leaves, stems, and roots were tested on the earthworms Pheretima posthumans. When compared to other extracts, the methanol extract of the root is the most effective and takes the least amount of time to paralyze and kill the worms. The crude alcoholic extract of C. ternatea, as well as its ethyl acetate and methanol fractions, cause considerable paralysis and death of the worms. In conclusion, the chemical constituents are well identified and understand from the previous research followed with their beneficial uses and impact on humans. The C. ternatea is a promising medicinal plant with a wide spectrum of pharmacological action that could be utilized in a variety of medical applications due to its effectiveness and safety. For recommendations, more research must be done to pinpoint the actual amount components present in the C. ternatea and to better understand the mechanisms of the plant's varied activities. Next, the presence of testosterone should be thoroughly investigated in order to gain a better understanding of the impact it can have when administered through the plant. Lastly, the actual amount should be discovered and prescribed because minimum quantity may have no impact or an overdose may have an adverse effect.